Kimberly Steele, and I am the director of the patient-led research for the Lymphangiomatosis and Gorham's Disease Alliance, which I am incredibly excited and happy to be here. And I've met many of you and look forward to meeting more of you as we come. So today, if my screen's gonna move, and you have to bear with me because this is my first time on Zoom hosting. So, Lisa, now my slides aren't working. Not moving forward. There we go. Okay. So today we are going to, the LGDA is presenting a 101 series, The Basics of Complex Lymphatic Anomalies. And why are we doing that? Well, we would like, as a lead up to our international conference that's coming up on October 2nd and 3rd, get our community prepared. And the subjects we're going to talk about are what is a complex lymphatic anomaly, the genetics, diagnosis, treatment, and research. And that's going to come over a series of 5 educational seminars, all aimed for our patients' families and also primary care. So invite your family doctor, invite your pediatrician, and see if they will show up. And that is to prepare us all for the LGDA International Scientific Conference coming up on October 2nd and 3rd, which we hope everyone will sign up and let everybody know about it so that we have a really good showing. Just first off, for anyone out there that is unfamiliar with complex lymphatic anomalies are a vascular anomaly. They are rare, and they include 4 subtypes. And you can see over here a drawing from one of our doctors and researchers in Japan who nicely schematics this with showing how they overlap. Okay, so that the clinical symptoms, the places in the body, and imaging features when you have an MRI or CT scan, the blood irregularities and complications overlap with each other, yet they are distinct. And what we're here today to talk about is that we do understand that these arise through what's called anomalous embryogenesis. And that means during development, when the fetus is developing, something happens to cells of the lymphatic system. So today, I'm excited to present our first webinar, number 1, and host it. And we are going to talk Genetics 101, and we're going to say, let's discuss somatic versus inherited mutations. And very, very wonderful to have today's speaker, Maanasa Polubothu, who's an MD, PhD, and I am going to let her take it from here. She is a pediatric dermatologist. She's from the Great Ormond Hospital. I always get this wrong, so I'll let you do this, Maanasa. And she is very interested in complex lymphatic anomalies, and not only is a clinician, but a scientist helping to solve some of the unanswered questions. And she's coming all the way from the UK, so it's late over there. And we're really excited and to have you. And I'm gonna let you take it from here. I'll stop sharing. And if you want to go ahead and share your slides, and I hope everybody can see the slides. Can someone give us a thumbs up? You can see slides. Great.
Amazing. And thank you so much for the opportunity to talk, and thank you for the introduction. So as Kim said, I'm Maanasa. I'm a paediatric dermatologist working in London in the UK. And clinically, we see a lot of patients with different forms of lymphatic anomaly, and we also do some work in our lab. So today we're going to do a very basic overview of genetics, and particularly the genetics that cause lymphatic anomalies. So this might be really basic for some of you. Some of you might have already know this extremely well, and for some of you it might be like a foreign language. So hopefully it will be interesting, and there'll be a chance at the end for you to ask some questions. So this is an outline of what we're going to cover today. So some very basic, what is a gene and what is a gene mutation? How do genetic mutations play a role in disease? The difference between somatic and inherited gene mutations, which is really key for understanding this particular group of conditions. We're going to review somatic mosaicism and talk about some of the factors that impact the clinical presentation of what you see. So starting with what is a gene and a gene mutation. So back to absolute basics. So the human body is made of 37 trillion cells. This is a cell here, and the center of that cell is the nucleus. And the nucleus is the place where the DNA is kept. So you might have heard of DNA before. It's our genetic, oh, sorry, I've lost my screen. Oh, sorry, I do apologize. I can't see anything.
Perhaps if you unshare and then share again, sometimes that.
We see the screen.
Can you? Sorry. Oh, perfect. I can see it again. Sorry about that. So each of your cells contains your entire genetic code, and your genetic code is enormous. So 6 billion base pairs, they're the basic building blocks of your genetic code. If you unraveled all the DNA in one cell, it would be about 6 foot long, and that's your entire genome. But because of a very specialized, extremely complex mechanism of coiling and folding, all of your DNA fits into these X shapes, which are chromosomes, and all of them fit into a tiny little cell. So like I said, your genetic code is made of base pairs or nucleotides. And this genetic language has 4 letters which code for every process, every cell, every protein in your body. So we can see them here. So A, G, T, and C. And everything has a genome. So bananas have genomes, all animals and plants have genomes. And the human genome is what we're going to be talking about today. So as I said, all of your extremely long genetic code, which codes for all the processes in your body, fit into these structures called chromosomes. And these are super tightly wound. So when people say DNA, they're referring to this helix structure. And this is a structure of the genetic code, so the joined up nucleotides, which is twisted into what we call a double helix. As you can see here, opposing strands. And twisted into chromosomes and twisted into your cell. So when we talk about gene mutations, we're talking about changes in these little nucleotides or base pairs, so these letters, this genetic instruction. So as I said, your entire genome is huge. It's 6 billion base pairs, and it's very difficult to visualize just how long that would be. And this is all the genetic code in one cell. So in London, if you ever visit, if you're ever allowed in again, or if you ever feel safe to travel again, in London, there's a building called the Wellcome Trust, and they have a free exhibit that you can go and see. And what we've done there is they've printed out the entire genetic code, and they've put it into 2 bookcases and filled it with books. So the book number corresponds to your chromosome. We have 23 chromosomes in each cell. So all the information in these 2 bookshelves is the exact code that we have in each of our cells, just to give you an idea of how long it is. And you might think, oh, maybe there's so many books there and 2 big bookshelves, maybe the font's massive. That if we zoom in, you can see it's teeny tiny. And again, it's just these 4 letters. So really the genetic code is a remarkable thing, and how it coils up into cells is a remarkable thing. But to have all this genetic code, every time a cell divides, and cell division is key to growth and development. So we all start off as 1 cell. We divide many times to become a human. As we said, 37 trillion cells. Every time a cell divides, all of this information in these 2 bookshelves has to be copied. So even with the most refined machinery, there's bound to be a few mistakes along the way. So you can see here, tiny little font. So just these are some basic principles that will just help you understand the relevance of our DNA code. So we have DNA in our cells, which is our genetic code. And there's two processes by which this DNA code becomes proteins. And proteins are important in your body for everything. So they signal, they communicate between cells, they instruct cells to do things like insulin and proteins. So your body depends entirely on proteins and cell signaling with proteins, and your DNA codes for this. So there's two main processes that happen to get from DNA to a protein. And the first is transcription, which is essentially making a mirror image of your DNA that can be read by protein making units in cells to make proteins. And proteins are essentially a series of different amino acids. So we don't have to understand too much about that, but that becomes important later when we're thinking about what the consequence of a genetic mutation is. So coming back to what is a gene mutation? So we know our genetic code is a series of these 4 letters, and is a set sort of series. So when people say, I got tested for mutation and I was wild type, or this patient was wild type for this mutation, what we're saying is the genetic code in that area is normal or matches the reference sequence. We can, however, when all the DNA is copying, have some changes, and common changes that happen are, so if we look at the top right first, sometimes when a DNA is being copied, you can have a deletion of a nucleotide. So when it's copying, it misses one out. At the bottom, you can see the protein that's been made directly from the DNA. So you can see that if we miss one of the nucleotides, we get a different protein further on. So that genetic change has led to a protein change. The other thing that happens is there can be an extra nucleotide added. So in this case, on the bottom left, you can see an extra U has been added, which again changes how this protein is going to be read from this DNA strand. And then the last thing that commonly happens, and this is probably what we hear about most, is a point mutation. So this is simply where one nucleotide is switched for another. So you can imagine having a different nucleotide might give you a different protein. So that particular mutation is leading to a change. So it's really just copying. So imagine if you were copying all those letters yourself by hand, it's either adding something extra, missing something out, or substituting something. And that's a gene mutation. So what's the role of genetic mutations in disease? So do all mutations lead to disease? So as I said, every time a cell divides, it has to copy miles and miles of DNA, because we're bound to make mistakes all the time, and that's true. So people have estimated between 3 and 50 mistakes are made every time a cell divides. So that's every cell in your body that divides. So we're all covered in mistakes, yet we don't all have lots of different diseases or disorders. So the majority of mutations we would expect don't lead to anything. So all mutations don't lead to disease, but why is that? So I said our genetic code is very long, but the genetic code is split into 2 parts. So you have your exon, which is your gene coding region, which is about 1% of your whole genetic code. So your whole long genetic sequence of letters, 1% is your exon, and that codes for genes. The other 99% is your intron. So that just means it's a bit in between the genes. We don't 100% understand what it does, but generally speaking, and to simplify this, a mutation in your intron doesn't usually lead to changes in a gene. So mutations, so for your mutation to cause a disease, it should be in that 1%, the exon that codes for genes. So if you do have an exonic mutation, so one of your mutations happens to have been in that 1% of your code, which codes for a gene, what can the consequences of that be? So we go back to this picture of our sequence. So in the top left, you can see our wild type sequence, or a normal sequence without any mistakes. In the top right, we see a synonymous mutation. So synonymous in the same way we would use synonym. So it's just a different word for the same thing. So in this case, what we have here, so if we look at this triple A in the middle, this was AAG, but it's become AAA. But both these nucleotide codes code for the same protein. So actually the consequence is the same. So we have the same protein at the end of the day, and we call that a synonymous mutation. So mutations that lead to the same effect, that lead to the same protein, don't cause any problems. We do, however, run into problems when we have a mutation that changes the sense of the genetic code. So, for example, here in the bottom left, this is a missense in which changing from this G to a C leads to a different protein. So we previously had a lysine, we now have an asparagine. So that protein now has a different amino acid in it. And depending on what that protein does, what particular role that part of the protein has will depend on how it affects the function of the protein. You can also have missense mutations. So that's again a mistake in a genetic code that lead to the protein stop being translated. So instead of coding for a protein here in the bottom right-hand panel, we've prematurely terminated a protein. So you'll have a protein that's too short. And again, depending on where in the protein that happens, depends on whether or not you'll have a functional protein, and that depends on and that will result in the consequence of that mutation. So not all mutations cause disease. Generally speaking, to simplify it, exonic mutations, so a mutation in that particular 1% of your genetic codes that codes for genes, will lead to something that might cause you problems. And even then, it has to be something that changes the structure of the protein. So hopefully you're all still following. So we've covered what genes are, why they're important, and how you can have gene mutations that lead to disease. We're now going to talk about a slightly complicated concept about germline and somatic mutations. You might have heard these words before. I was saying to Kim before we started, actually, sometimes doctors who aren't familiar with this particular group of diseases won't be too familiar with the concept of somatic mosaicism. It is quite not a new concept, but something that we've certainly learned a lot more about in the last 5 to 10 years. So I'll go slowly, but there will be a chance to ask questions at the end if I say anything that doesn't make sense to you. So we've talked about mutations, and mutations happening in cells as they divide when they're copying the genetic instruction. So we talk about germline and somatic mutations, and the key difference between the two is the timing of the mutation. So if you think we're all formed by a single cell, so a sperm and an egg join in fertilization to make a zygote, and that's the first cell. And then that cell divides millions of times to make a baby. If your mutation is present in the first cell, or something that's passed on from your parents in either the sperm or the egg, we call this a germline mutation. Because it's present in the first cell, it will then go on to be in every cell that divides from that cell. So it will be in your germline in all of your cells. A somatic mutation is something that happens after this first cell stage. So we sometimes call it a post-zygotic mutation. So it happens at some point later. And I'll show you examples of how that means it's not going to be in all of your cells. It's only going to be in a number of your cells. And really that depends at what point it happens. But really the key difference between somatic and germline mutations is timing. So something that's present in your first cell is a germline mutation, and then you have it in all your subsequent cells. Something that happens later on in development after that zygote stage is a somatic mutation. So this is just to summarize it again. So germline mutations, these are present in the first cell, therefore in all the cells of your body, and these are the type that can be passed to offspring. So typically speaking, when people talk about genetic disease, they tend to be talking about germline mutations. They tend to be talking about things that can be passed on between parents and children. So things like cystic fibrosis, things like neurofibromatosis, lots of other diseases that you would have heard of. That's the majority of disease, probably that's discussed when you think about genetics. But somatic mutations are these mutations that happen after birth. So because they're not in that first cell, they occur later on during development. They're not present in all the cells of your body. Depending on when exactly that little mutation happens will affect how many cells are affected in the end. So if it happens early on, when you're When you've just started dividing, it'll be in lots of your cells. If it happens super late in the third trimester, when you're almost finished developing, it will only be in a small proportion of your cells. And generally speaking, to simplify it, somatic mutations cannot be passed on to offspring. You shouldn't have them in your gametes. They weren't in your first original cell. So when we talk about mosaic disease and somatic disease, which covers GLA, lymphatic anomalies, other types of overgrowth and vascular malformation, we're generally talking about somatic mutations, which we don't think can be passed on to offspring. So just to summarize, so this is a classic picture of germline mutations. So we all have 2 copies of each gene. We get one from our mum and one from our dad. And then we can pass those on to our children. So this is, so you can see dad's got 2 purple genes, mum's got a purple and a green gene. The green gene is the affected one. She's either going to give the green or the purple to her kids. So she has a 50% chance of passing this on to her kids. So that's typically your germline mutation. And when she passes it on to her kids, they'll have it in all of her cells. They'll have 2 copies, mum and dad's copy, which is why it looks like half and half, but it'll have it in all of her cells. So that's germline mutations, and that's a bit more simple. So we're going to review somatic mosaicism. I'm going to say the same thing in a few different ways using a few different pictures, because I do think it's difficult to get your head around because it's so different to what we're typically taught in school and in college about the way genetics and the way things are passed on. So mosaic disorders, so the definition of a mosaic disorder is the coexistence of 2 different genotypes. So genotype is what your gene is, so you can be wild type or have a mutation, in a person derived from a single zygote. But I said to you before that every time a cell divides, there's at least 3 mutations. So we're all littered with mutations. But I also said to you that the majority of those mutations won't do anything. They'll be in your non-coding DNA, or they'll code for the same protein. So by that definition, we're all mosaics. So the definition that we're now moving towards for a mosaic disorder is the coexistence of cells with at least 2 different gene populations by the time you're born in an individual that started off as 1 cell and which something that leads to a disease. So that's what we're thinking of when we're thinking of the group of disorders with a lymphatic. So we're thinking the original cell didn't have a mutation, somewhere during development something happened, and then a disease phenotype was evident at birth. So just a different way to illustrate this. So we all start off as 1 cell like this, and then that cell divides, and then each of those cells divide, and then each of those cells divide. So it's exponential. At some point during division, there was a little mutation shown by the green cell. And all the cells that then divided but were created by that cell dividing have a mutation. So we can see we have lots of cells and a proportion of them have a mutation. So we all start off as a ball of cells, and then we very cleverly unfurl to be a human body with arms and legs. So just thinking about mosaicism not in a ball shape. So we start off as 1 cell, we're dividing, we're dividing, we're dividing. A little mutation happens at some point, then all the cells that result from that original cell will have a mutation. And when we think about some of the mosaic diseases we see, so vascular malformations, overgrowth syndromes, lymphatic malformations, it sort of makes sense with the distribution you're in. You can think something happened here, and then all the cells in this area have come from the original cell and look a little bit different. So as I said before, the timing is really key. So 2 different people can have the same mutation and have completely different disease. Someone can have very mild disease and someone can have really extensive disease all over their body. And really it's all about timing. So when did that mutation happen? So this is a ball of cells starting off from 1 cell. If you have a very early mutation in the first few cell divisions, it's likely that that won't be supported. So the majority of mutations that we see in overgrowth syndromes, vascular anomalies, lymphatic disorders are really strong mutations. They're mutations that we often see in cancers. So if that happened too early, that wouldn't be supported. And it would most likely result in a very early miscarriage. If it happened a bit later, but still quite early, still in the first number of cell divisions, you would get a very extensive phenotype. So I'm using the example of a capillary malformation, which is a type of vascular malformation, which is a pink stain, just because it's quite easy to see. So this little change in the blood vessel cell that led to this capillary malformation happened early on in the first few divisions. And you can see this child is severely affected. It affects a large proportion of the body. If it happened a bit later, then it might just affect a single limb, a single digit, maybe a head. And if it happened super late when the baby was almost completely formed, so the same mutation, same cell type, you might just get something very simple, like this very simple capillary malformation of the hand. So we know timing is important. So what you'll probably hear at the conference next week, what you've probably heard before is phenotype. People use phenotype a lot. And phenotype just means what you can observe. So we like to talk about genotype and phenotype. Genotype is what your genetic mutation is, or if you have a genetic mutation. And phenotype is what we can see. So the extent of your disease, how severe it is, where it is, what it affects. So we've already said timing of a mutation can affect the extent of a disease, but there's lots of other things that also affect mosaicism. So one of the other key things that affects the extent of your disease in a somatic mutation is the cell type affected. So if you think we all start off as one cell, but those cells then go on to divide to make up all the cells of your body. So we have fat cells, we have blood vessel cells, we have lymphatic cells, we have pigment cells, we have many different types of cell. So if your mutation affects a very particular type of cell, so a differentiated cell, so a cell that's already gone through the process and now can just be a lymphatic cell, that's all it can be, then you'll get disease that only affects the lymphatic cells. If it affects a cell higher up, so a cell that has the potential to go on to be a lymphatic cell or a blood vessel cell, you might get some lymphatic disease and also some venous disease. So that's a very simple way of looking at it. But really, if you have late differentiated cells that have already decided they're going to be a lymphatic cell, a fat cell, etc., it will just affect that cell. But if it happens earlier on, then it has the potential to turn into lots of different cells. So that's one other consideration. And other things that affect the phenotype of mosaicism, sometimes some of them we've spoken about. So what's the normal function of that gene? Is that gene important? Is that gene expressed in the cell type that developed the mutation? So some genes aren't expressed in lymphatic cells, for example, so a change in that gene wouldn't make a big difference. The exact mutation, so we know that the gene code for each gene is very long, but different parts of that genetic code will be key. So they'll code for important parts of the protein that are really critical to the function of that protein. And other parts of a gene might be near the end and might not be as useful. So the exact mutation, where it is, and also the background genotype. So some people who get a somatic hit will have a predisposition based on their other genes to develop different aspects of a disease to have it more severely, if that makes sense. So we're almost there. So you've heard people talk about your clinical diagnosis and your genotype. So what's the relationship between your clinical diagnosis and your genotype? Is it very straightforward? If you have one clinical diagnosis, then this is your gene, or does one gene only lead to one clinical diagnosis? So I'm sure all of you know that that's not the case. It certainly isn't the case with our group of disorders. So it's important to understand that the same clinical diagnosis can result from lots of different mutations. So we know, for example, GLA, we know some of the causes of GLA is NRAS, and we know some lymphatic malformations are caused by PIK3CA, but there's other genes that we don't know about. There's other children who don't have an identified mutation. So different genes are causing the same clinical diagnosis. So an example of this is capillary malformations, which again are those flat pink stains that I showed you, and they can be caused by a number of different genes. So shown here, PIK3CA, which you may have heard of, GNAQ, GNA11, which are in the same pathway. So same phenotype as a result of different mutations. And then conversely, the same mutation can lead to different phenotypes. So the exact same genetic change, sometimes in a similar cell type, can lead to lots of different clinical diagnoses. So for example, PIK3CA. So we know a mutation in PIK3CA, it can be the same mutation, can lead to overgrowth syndromes, it can lead to simple venous malformations, lymphatic malformations, and then a mixture of all of these, and lots of old diagnoses that we use, clinical names such as Cloves, Klippel-Trenaunay. You might have heard of some of these. We know can now be caused by PIK3CA. So same mutation, same gene can lead to different clinical diagnoses. So at the moment, these are the genes that we know about for sporadic and mosaic vascular malformations and lymphatic malformations. So lots of genes. Some genes cause more than one condition. Some genes, some genes overlap. And again, like I said, it's the same mutation. What's interesting about this family of genes is they're all linked really in the same sorts of pathways. So all of these genes code for, all of these genes code for proteins that are part of key signaling pathways that are important for your growth and development. Here we go. So these are some of the pathways that are involved in the generation of lymphatic anomalies. So the main two are your MAPK pathway and your PI3K mTOR pathway. So these genes are all coding for proteins in these pathways. But the important thing about this, and the whole reason that we go looking for genes and we spend so much time talking about them, is medicines are being used against particular proteins. So knowing your gene mutation might give an indication of what medicine might work for you. So though lots of children can have the same clinical diagnosis, they might have different genes and they might respond better to different medicines. And these are some of the medicines that I think you're going to hear about later in this seminar series, along with some updates on some new medicines that people have been using recently. So in principle, your gene diagnosis can predict what medicine will work for you. So that's why it really is important to understand your genetic diagnosis. So depending on where you live, you might have different availability to genetic services. But I think part of this network and part of the beauty of this network is you'll be able to get in touch with someone, or you'll be able to help your doctor get in touch with someone who might be able to help you get a gene diagnosis. Because as I said, this is going to be the mainstay over the next few years for helping choose medicines. So just to finish, it's important to know both your clinical diagnosis and your genotype so we can help choose treatments. And that's why we've been talking about genes today. So to summarize, before we open for questions, genetic variants are common events that occur in every cell division, but in certain situations, a genetic change can lead to disease. These changes can be germline, so present in the first cell and inherited, or can be somatic, which means they occur spontaneously during development and are sporadic, i.e. generally not passed on. In mosaic disease, the phenotype depends on a number of factors. The main ones are the timing of the mutation, the cell type the mutation affects, and the type of mutation. So understanding these gene mutations is really key to us understanding the process of a disease, and designing new medicines and deciding which people will be most suitable for which medicines. But you'll hear more about that later this week. Thank you.
Wow, that was wonderful. Thank you so much. I am just really taken by how well that was explained in an easy way, and thank you. So our bandwidth is a little rough here. I think we've got a storm coming up, but everyone stay with us here. And as far as questions and answers, we would like to open it up to everyone here now and ask any questions. And until any come in, maybe I'll start. As I hear Dr. Polubothu, you speak about the importance of a clinical diagnosis. Can you explain, because I think many of us don't even know, I think it was really important that you brought up that we have to talk to our doctors and ask them about that opportunity. So when we do go to the doctors, how do they clinically diagnose a patient with a complex lymphatic anomaly? In our case, in our son, we knew that we had to try and get tissue, and that's dangerous. We tried tissue and we didn't find anything. And so maybe you can help us learn more about how you go about the clinical diagnosis and how to talk to your doctor about it. I've met some in our community that have tissue and it's just in storage. So when I hear that, it's like, we need to get it to an expert. So maybe you could tell us more.
Definitely. So I think there's two reasons it's difficult. So I think the first is we're dealing with a group of diseases which are extremely rare. So whereas we see them, on a weekly, monthly basis, your average pediatrician, your average family doctor will maybe see one in a lifetime. And when things are extremely rare, you'll tend to look something up. And I think you'll all know, perhaps from researching things, the internet is full of myths and misconceptions and lots of old terms. And then the second reason I think for this group of disorders, it's particularly hard to get a clinical diagnosis, is that what we know has really changed in the past 10 years. So the way we label things has really changed. So before we had lots of clinical terms. So if I use pick-free-CA overgrowth as an example, because that's probably an example of where there's so many different labels. So if some people would be labeled as CLOVES or KTS, or depending on where your overgrowth was or what it looked like, you'd get a different label. And all these people thought they had different things. But really now we understand that these are all a spectrum of disease caused by a particular genetic mutation, and it's all to do with mosaicism. So it's really difficult to get a clinical diagnosis, and it's really difficult. But I think you shouldn't have to do, but I think what I've heard of a lot of families doing, advocating for their children. So finding support networks like this, finding experts, getting in touch yourself, asking your doctor to get in touch is probably the best way to go forward. I don't know if you'd agree, Kim, having different experiences. Oh, sorry, Kim, you're muted.
Sorry, I got to get used to this hosting thing. Does anybody have any questions at all? Because now's your chance. I don't see any yet. Put them in the chat for us. And if there aren't any others, I still have some. And
I've got one here from
okay.
from all the way a fellow Scot. This is very, again, that was a super interesting, and very clear, how you went through that. My question is probably more individual to me as opposed to a universal question. Probably more. I have developed a condition called chyliocystitis after a surgery last year. So I think we're going down the genetic route now, but what are the chances of there being a genetic mutation presenting itself later in life?
So if you have something on this in the spectrum of disease, the mutation will always be there. So it will be there from birth and it will still be there later in life. So you can really take tissue at any point in your life. If you take the correct tissues, you have to take the tissue that's different, which can be tricky. And you can get a genetic mutation. And if you're based in the UK, that should be reasonably straightforward now.
I think
where you live.
I think it's more I was absolutely fine until I got to 32. I never had any medical issues, and then after having a surgery, I developed chyliocystitis, and we can't work out why it's happened. We can't find a lymphatic leak is
Wow.
happen. And
We're having trouble hearing you. Maybe what would be great is if you want to contact Michelle, if you want to contact the support line or my email, we can try and answer some more questions for you, because the bandwidth isn't so great.
yeah, that could you pop your email address in the chat, and I'll send across an email. Thank you so much.
Okay, and so just moving on, Dr. Polubothu, we've got some questions coming in now, which is terrific. One comment from Elizabeth Manty, she noted, and I think it's very important, I also think so often the first doctor will want to get a biopsy, not realizing that for this patient population, it can be dangerous. And so I think that's a really great point, because again, in talking with the community, oh, could whoever is on mute for us? Thank you. In talking with the community, what I have learned from them and sharing, and even from our own experience, that sometimes the biopsies are not done the right way, or as Elizabeth said, it can be dangerous. So I think one thing I've told the community is make sure first that the doctor who's going to do this biopsy has done these before, explains it really well, and that an expert from a vascular anomaly center is involved. And maybe you could comment a little on your thoughts about it. Oh, the bandwidth. I think we've having trouble.
Did you mean?
with- Okay, there you go. Oh, you froze again.
Did you mean my comments on it?
Yes, yes.
I think the transatlantic connection's giving up, isn't it?
I know.
Yeah, so it's very difficult to, so absolutely. So it might not be safe in that group, but also if you're going to go ahead and have a biopsy, you really want to get as much information from that biopsy as possible. So you want to have, if possible, tissue from genetics, tissue for histology. You want it to go to the right place, be fixed in the right way. So you're right, jumping in is definitely not the thing, but I think again, using the network to find an expert at a vascular anomaly center and seeking their advice before anyone jumps into anything, as Kim said, is really good advice.
All right, the next question is, how difficult is it to find the mutated gene? Can the testing be inconclusive?
That's such a good question. I'm really glad someone asked that. So absolutely. So the difficulty with Mosaic disease is your mutation only affects a number of your cells. So depending on what, so for example, if you take a sample, a biopsy, a normal biopsy, a little bit of skin, you'll only have a few lymphatic cells in there, for example, if it's a lymphatic disease, maybe 1%, maybe 5%. And the majority of, you need to use targeted deep sequencing, so a special type of sequencing to pick that mutation up. And then there's still a chance you might not pick it up because the number of mutated cells in a biopsy is going to be very low. Compared to if you had a germline disease, you could sample any tissue in your body and it would be in all your cells. So it's sometimes you get a negative result, but that doesn't mean you don't have a mutation. It might mean that there wasn't enough. So lots of work is being done on refining the different genetic processes to maximize our pickup on samples. But it is a very good question, but it's something that you should know before you have a biopsy. There's a chance it will be inconclusive and you might not get a result from that first test.
Yes, that's exactly what happened in our son's case. He went through the biopsy, but it wasn't enough tissue to pick anything up. And so now the question is, when do we try to biopsy again, or do we? and we knowing how dangerous it is to get tissue. And right now he's very stable. So some parents, patients also are concerned, when do we do this? When is the timing, the right timing? Because we don't want to rock the boat if the kids or the patient are stable. And I don't know if you have anything that you can tell us about that, but that seems to be a common question amongst the community, like when do we do the biopsy? We're having bandwidth.
I'm so sorry,
Maybe if you go off video for a bit, it might help your bandwidth.
You can't hear me. So it's a really good point.
It might help your bandwidth.
I'm so sorry. I really hope you can hear. So it has to be a decision, and you have to weigh up all the benefits. So actually having a biopsy and knowing what it is and knowing what the gene is might mean that you're eligible for a medicine that could keep things at bay. Also, some of the medicines we use, like sirolimus, wound healings, if you're starting something like that, it's probably worth getting a biopsy beforehand. And again, it really depends on preference, on how confident you are you're having a biopsy done in the right place, but hopefully you'll be able to be guided by your local experts as to when. But there's no clear answer, and every patient is so different, as we, these mosaic conditions.
And now I just picked up some more chat, and I thought this was excellent from Elizabeth Manty. She said, if you're doing a surgery for any reason, have them take a little extra if it's safe to store and use. And I think that's a great point. I don't know if you want to say anything about that, but I tried to give that advice too. Hopefully you don't need surgery, but certainly if you're going in for surgery, if the doctors can get tissue for research, it's a good thing. Can you also, I think, just going on from there, the fact that tissue diagnosis is the way to go right now, but dangerous. Can you tell us a little bit about liquid biopsy and the hopes for that? I know you said that there's technology is advancing. I think everyone, we may have lost Dr. Polubothu. Oh, let's see. Well, let's see if she comes back, everyone. It does look like we've lost her. Maybe everyone in England is. I see Stephanie, do you have any ideas? Oh, here's, no.
I'm in England and mine's okay.
Mine too. Huh. We lost her for right now. Oh, here she comes back. Hopefully. Maybe. I think we're having definite bandwidth problems, so I apologize, everyone.
I'm so sorry, I was temporarily exited by trying to get her.
We thought it was bandwidth, so glad that you're back. So I was just asking if you could comment a little bit. We know tissue diagnosis is dangerous, but essential because we need a target for medical treatment. Are there, you spoke of new technologies, are there some new technologies on the horizon, for example, like liquid biopsy that's done in cancer diagnoses? And maybe you can explain what liquid biopsy is. I guess I'm using a big word there.
Oh, so there's different ways to get your mutation, but people are trying lots of different things. So some of the things that might be relevant and might have obviate the need for having actually a biopsy. So is getting DNA from different tissues. So one of the things that's new and seems to be routinely done for different disorders is, so you're circulating DNA in your blood. So you can take blood DNA and siphon out cells from an area and try and test for that. And then the other thing done in lymphatic malformations is taking fluid from cysts and extracting DNA and trying to get a mutation from that. So at the moment, these things aren't routine. At the moment, your tissue diagnosis seems to be the most accurate way to get a diagnosis, but certainly in the past few years, it's come a long way. So recently I've seen talks saying they can detect maybe between 50 to 80% of known mutations using cyst fluid. So these are all things that hopefully in the next few years will become standard practice, because obviously that would be a very safe option. You could do it early. You wouldn't have to worry about, you could do it again if you needed to, if you didn't get enough of a sample the first time, and it would avoid, multiple biopsies or a biopsy if you weren't having a surgery.
That's great. And I think one message is when our community is having a procedure, it's okay to speak up and ask, should they be collecting the effusion? And should it be sent to see if they can find a mutation? It might not be possible, but I think using our voices and asking is a really good thing. I'm making the comment now, right? So as a parent too. There are some questions here. Do we have information? This is from Anila Morina. Do we have information on which genes have mutations in GSD?
So I think there's some, so in the research community, I've heard different murmurings over the years of different possible genes. It may be genes, so at the moment there's nothing, as far as I know, that's been published, but I have heard different murmurings of new genes that might be or have been found in a small number of patients. So again, that's something that I think in the next few years we'll know a lot more about. And then hopefully again, hopefully it will be the same sorts of pathways and it'll be drugs ready to go.
Just to give some hope out there, we have some terrific researchers. Our very own Dr. Michael Dellinger at UT Southwestern is studying GSD very, very strongly. So, and hopefully with more collaboration again, internationally, we can really pick up and advance the science. I did hear that possibly, I think it was KRAS might be implicated in GSD, but again, they haven't targeted one gene yet. This is a comment from Mary Kinry, if I said that right, I hope so, Mary. And I think it's a good one, a question. When you find a mutated gene and link it to a targeted therapy, will that therapy always have a positive impact on the disease?
So not necessarily, and I think what we're seeing in all of these complex vascular anomalies is, so the typical way to study a drug and to know if a drug is safe and effective is to do a very large clinical trial. So for something like diabetes, that's very easy. You can really standardize it. And also because they're not mosaic diseases, the diseases all present in a very similar way, similar sort of severity. But for something like this, which is so unusual, and when we do get patients, they really need treatment urgently often because their disease has progressed so much. We tend to use compassionate use drugs on a named patient basis. We're not able to do as much as we normally would, where we would choose a dose, and often because we're using it in a situation that's already very critical, it's difficult to see how much of an effect it's had. So I think based on case reports I've seen of different drugs for different things, sometimes drugs are used and they conversely make something worse. And it's just one of those things that we don't fully understand. With all of these pathways, there's negative feedback. So if you inhibit one thing, you might overexpress something somewhere else. So there's always the chance that you might make something worse, or something might work for a while and then something else might be overexpressed because you're knocking something down. So it's very tricky. At the moment, it's our best bet. And I've seen lots of case reports of very successful use of targeted drugs, including in GLA. So it's just a case of learning patient per patient. And yeah, because it's not as straightforward as that, unfortunately.
I know. I think you made an excellent point, though, to all of us here today, that I have, as a parent, and now coming into this this community, is the importance of participating and sharing our experiences, because it is rare. And it's not like diabetes, so we can't do those clinical trials. And so we are looking for other ways. And some of them are drug repurposing, and also collecting data and things like a natural history study or a registry, where we follow our patients a long time, over time, I mean, so that we can understand the natural history of the disease and look at the medications we are using. So thank you, that it's such, you hit on such an important point. The other thing I think that's super important is, now I'm forgetting. I had this idea, and that's like, oh my goodness. Well, I'm not sure. Dr. Polubothu, anything else you want to say until it comes back to me? I'm getting old.
No, just thank you so much for your questions. But really, this research network, it's just so fantastic. And I think to have this support for patients, I was saying to Tracy, I had a patient who was quite unwell. It came about 4 weeks ago, and I was able to tell her about the patient support group and the other things. And she, the family had been told by different doctors they'd never seen this before, and it was almost certainly this, and it was almost certainly that, and lots of misinformation. I think just having this group and this resource to direct patients to, I found really valuable, and I think must be such a big reassurance. And all of these events, I'm sure, will be really well received. So yeah, congratulations to all of you. You're doing such a good job.
Awesome. The last comment here is lovely, from Elizabeth Manty, from a parent who's been in the community for 14 years. It's getting better. It was so hopeless then, and now hope is on the horizon, and there are amazing doctors and clinician scientists like Dr. Polubothu, who are out there working 24/7, and they're going to find the answers. I think it's fantastic. So if there are no other questions, just to bring up that, and we thank you so much for your time. I'm hoping we can get you more into the community now, Dr. Polubothu, and network and learn from each other with different information that we get from each other. So with that, just to end with the fact that this was one of 5 in a series that we're really excited about in the LGDA. Our next webinar is very close, coming up on Monday, and it will be held on August the 9th at 12 p.m. Eastern Standard Time. We hope that you'll all join us. Thank you for giving up your Saturday. Dr. Polubothu mentioned that at the beginning. She was amazed at how many of us showed up, so thank you. And we hope that you'll attend these seminars so that we all learn from each other, and then hoping that we'll see you at our, well, first of all, let's talk about the next webinar since I made the slide. So on Wednesday, August 25th at 1 p.m., we're going to talk about preclinical models. And that's going to be important when you attend the conference, so that you are able to follow the basic science, which sometimes can get very much over everyone's heads. And so they will, Dr. Dellinger and Dr. Lacroix, who are both basic scientists, and again, doing incredible work in GSD, GLA, and KLA, will talk about preclinical models. And that's the mouse model, the animal model, in vitro, which means in a dish, and in vivo by using different tissues. So that's going to be really a great talk on Wednesday, August 25th. On Monday, August 30th at 12 noon, Eastern Standard Time, we will talk about complex lymphatic anomalies, just an overview for the layperson. What does it all mean? And that's going to be presented by Dr. Manuel Carcale from Toronto Sick Kids, which is great. We're being very international. And then in probably the second week of September, we haven't nailed down a date yet, but it will be announced. We are going to do treatments, and that's what Dr. Polubothu had mentioned when she showed us the pathways and that there are targets and the medications will be targeted. So we are going to have Dr. Kristen Ricci from the Cincinnati Children's Hospital give us an overview of the current and possible future medical treatments for complex lymphatic anomalies. And then, if my slide will work, sorry, I know, bandwidth, I guess. So I do have another slide, but it's not coming up, which figures. Hopefully Monday, it will go smoother, everyone. But in the meantime, the LGDA International Conference is on October 2nd and 3rd. You can find a link to register both on the website at lgdaalliance.org or through Facebook. And we'll also post it on some other social media areas like Instagram and Twitter. And so, since my slides will not advance, I think we will call an end to our first webinar. We're almost on time. Does anyone have any questions? Shout them out, because we're here now, maybe quickly that we can answer. And anybody want to say anything? Give you a chance. It's quiet. Okay. I think that will be it for today. And thank you for being here. Thank you for allowing me to try to host. We'll try on Monday again. And reach out. Please feel like you can reach out, ask questions, and remember your voice is the most important. And that's how we're going to change things. So thank you for all being here. And Dr. Polubothu, that was awesome. Thank you. Take care, everyone.